Awesome. Awesome. Uncle Robert, how's it going?
Well, it's going fine.
Fantastic.
I'm a free man. I just retired.
Oh, officially. That's wonderful.
Yeah.
Wow. That's terrific.
Now tell me what I can do to stay amused.
Yeah. Awesome. So this is Sam Alexander. He's a mathematician friend of mine. And then I see someone else has joined. Hey, Dave Gorin. Awesome. Hey, man.
Hey, friend.
Right on. How's it going?
Good to see you.
Terrific. Thanks for joining. Boy, this is exciting. Well, it's 10 o'clock, so I guess let's get started. I'm dying to hear y'all's questions if you have any that you've sort of thought of. I was telling Drew a minute ago, most of y'all have heard some version of this idea from me in person over the past 4 years. And so, those 3 lectures that I just published are kind of trying to wrap up loose ends and give a more thorough account of it all in one place. But because, long essays that are on Medium or on my website are hard for people to get through. But yeah, I'll just open it up to questions right away. I've got a question from my dad to start with if any of y'all don't have a burning question right this sec. Okay. So Luther Munford emails in to ask if traits are passed on by something other than genes and that something is determined by oscillation, shouldn't there be some biological evidence of it? Yes, I think the answer is yes. And the something other than genes is cells. So inheritance works not just by passing along genetic material, but by there being an entire cell that's involved in inheritance. And that's where epigenetic inheritance passes through. And so the question is, if epigenetic inheritance is widespread. So, and just to be clear, my understanding of epigenetics is that it's, conventionally considered that the mechanism of the cell interprets the genome, right? It tells what the mechanism of the cell decides what genes to turn on and off, and that determines differentiation and the rest of development. And hey, Barrie.
Hi.
Morning. We're just getting started with a question from my dad that's posted in the chat here. Right. So the answer is the whole cell, and the question is what makes the cell operate, right? Because if epigenetic changes can be inherited indefinitely, and it seems like in at least some cases they can, and it seems like to me the more we look, the more cases of this we find. If they can be inherited indefinitely, then it would be like a game of telephone where the instructions are one thing and the interpretations are changing, right? And if they're drifting randomly, then the instructions would no longer work. They would become dysfunctional. And so the question is, what's keeping them functional? If epigenetic changes are being inherited indefinitely, what makes those changes functional? So yeah, does that answer the question for anyone?
Charlie, I have a question. So if the epigenetic characteristics of the cell are inherited, they would have to double, wouldn't they? They would have to copy themselves in some way. How does that happen? You can't inherit something through several generations without increasing the number of those molecules somehow. Or do I not understand?
Well, just when the cell divides, right? I mean, when the cell divides, it self-templates.
Yeah, but if those factors aren't copied, when the cell divides, there'll be half in each division, right? So that may not be enough to continue, and then you go several divisions and the molecules are no longer there.
That's a great point. I think the way I think about it is not as molecules, individual molecules, but as processes. And that's why this idea of oscillators to me is explanatory, because we're asking the question, how does the divided cell recreate the same processes on either side of the division? And you're right, it has to absorb new molecules and put them into the same sort of dance to make a functional cell on either side of the division. And so I think what I'm like understanding from your question is there's a specific way that genes are copied, right? That DNA is copied, and we can see the particulate inheritance on either side of the cell division. And so we can say, okay, this is a haploid gamete on this side and another haploid gamete on this other side. And for those non-biologists, Uncle Robert is like a very hardcore, lifelong esteemed biologist, guys. So I'm trying not to get super like technical. And I'm not, obviously. So but to me, the question is, I mean, your question is what carries that information if it's not a particular molecule? And if it is a particular molecule, how is it copied?
Right.
And that's the mystery, right? What carries the information? Because there's a process. And so to me, the idea of an oscillation is that, you know, you've got an ongoing process that carries in itself sort of the seeds of its own recurrence. So, but correct me if I'm wrong, the concept of epigenetic inheritance is not really controversial anymore. I mean, it's wide, it's been found in many organisms, including humans, right?
No, you're teaching me something. I don't know that.
Okay.
But go ahead.
Okay. Well, I think that that's my best attempt at an answer to that question. I think it's really, is everybody clear on the idea of the game of telephone? The reason why, if epigenetic inheritance is widespread, that poses a serious problem for the genes first theory.
Flesh it out for me. Yeah.
Okay, yeah, because this is like kind of the heart of it's possibly the strongest proof for the idea that intelligence could be a leader and genes could be a follower in evolution. So the idea is, if the genes are instructions, right? You have a list of instructions, and then you have the cell, which is an ongoing mechanism for carrying out those instructions. And there are many ways to carry out the instructions wrong, like the wrong way, right? So there's 30,000 genes, and if you express the wrong genes at the wrong time, the cell will just disintegrate. It won't stay alive. And if just computationally, if you think about the number of ways that 30,000 can be uniquely combined, it's a big number. It's bigger than the atoms of the universe. And so there's got to be something that's telling the cell which genes to express at which particular times, right? And so that's the sort of interpretation in this script here. The normal view of life is that there's instructions in the genome, and then the cell is just carrying out those instructions, right? So you with me? Okay.
Yeah.
Right. So if the cell starts carrying out the instructions in a different way, there could be like that theory could hold up. If the cell just carries it out, there's some variance in the way that the cell interprets the genome, and it's harmless, right? It just varies a little bit and then it kind of regresses back. Like sometimes sometimes a cell will make a little bit more of this protein, sometimes it'll make a little more of that protein, but on average, it all works out because the genome is what's telling the cell to behave correctly. Okay. But if the cell can start interpreting those in a different way systematically, okay, it can change its interpretation systematically. And then those changes can be inherited indefinitely. You with me? Then you've destroyed the whole logic of evolution, because you can't, there's no reason then why that interpretation wouldn't just drift randomly into a totally dysfunctional pattern. So there has to be something that's keeping that interpretation functional if it's inherited generation after generation.
Quick question. What's the role of context? If it sounds like epigenetic inheritance, if I'm hearing you right, it's you're saying that there's a particular interpretation of the genes that the genes are not themselves responsible for, that determines how genetic expression happens, and it's not the genes themselves; it's something else. Right. And that this interpretation can be inherited from one generation to the next generation.
Right.
It occurs to me that one, I guess I'm thinking two possibilities. One is that there's something in the organism, in like the network, that is passing itself down as an interpretation that's not genetic, but is conditioning the interpretation of the genetics. And or there's something in the shared context between generation A and generation B. For instance, the context of a woody area in Mississippi, for instance, and that context is actually influencing the interpretation one generation after another. Does that when I say context, do you understand?
Well,
that's brilliant. Yes, that's brilliant.
It has to be the environment, right? It can't be embedded. If I understand your theory correctly, Charlie, it has to be the environment interpreting the genes.
Right. Exactly. And so like the most graphic example of this, I mean, you just ask the question, what causes a stem cell to do what it does, right? So everybody, like every organism, multicellular organism, begins with just one cell, and that cell is, totipotent stem cell, and then it turns into all these other lineages of cells, and each lineage of cell differentiates into a totally different structure, like bone or brain or skin or whatever, and all of them carry the same genome. So they carry the same, like if the genome were instructions, they carry the same instructions, but the context of where they are and what's happening to them around them causes them to differentiate. And not just that, but it causes some cardiac cells to behave differently than other cardiac cells based on their position in the muscle and so forth, and same with the skin and everything. So it really determines this profound, these like very profound changes. And yeah, so I'm trying to like strengthen the genes as instructions view as much as possible before like just disagreeing with it, because I think that the standing idea is that the genome sort of tolerates all of that, that it's capacious enough in the way it prescribes things that it prescribes all of those changes, and then it tolerates some variation from the environment, and that's all fine and dandy as long as those changes the changes in the process of development and speciation and interpretation of the genetic code can't be inherited. But if it can be inherited indefinitely, then we have to explain how that results in a functional system.
So Charlie, when you speak of inheritance of extra genetic, extra DNA changes, patterns. To David's point, like what do you think is the mechanism that drives that? Is it the environment? The way I read it, it seemed like it could only be the environment. Do you think there's a different way that it happens?
Right. No, exactly. Yeah, it could only be the environment. And I mean, on the local level, it's the environment immediately around the cell. It's what's happening immediately around the cell. And obviously that scales up to the whole organism. If you want to explain what someone's doing, you have to.
Yeah. Well, so you're, I mean, in a sense, you're flipping the classical story on its head, in that instead of being driven by the genome and DNA, it's being driven by the environment primarily, or at least there's much more of a balance between the two.
Right. I mean, I'm definitely not saying that genes have no effect, or that genetic inheritance is not important. Like it's as if, like, my metaphor for the whole thing is tools in the toolbox.
Well, yeah, but I think maybe what David and I are trying to say is, like, just state that clearly.
Yeah.
Don't be afraid of saying, like, that's what I'm trying to do.
Well, and I hear, Charlie, in the way you're addressing Andrew, your read of context is really cool, in that it's intra-context. Your sense of context was the context for the cell are the cells around it, which is like that is the context for the cell. I think when I was using the word context initially, I was thinking about like the context outside the organism. And there's no good reason why that context should be context, and the context for a cell inside an organism isn't the context. I think they're like different degrees of outsideness. They're different degrees of context, I guess. But I'm curious what you think about. I guess I'm wondering if you want to lean on the way that cells around one cell are the context, as opposed to Mississippi or the forest. Does that make sense?
Yeah, it does. I mean, you have to, like, in order for it to be persuasive, it has to be specific as possible, right? Because there are many organisms in Mississippi. I mean, that's like too big of a container in the HSD framework. But it's yeah. So I mean, one way I like to imagine, like, one thought experiment comes from Dennis Noble, who is Uncle Robert's tutor at Oxford, who's this fascinating biologist who writes about this, complex systems, and he initially did most of his early work on cardiac cells and cardiac rhythms. And one question he asks is, why is it that two adjacent cells in the same tissue, you look at the expression rate of a given protein, a particular protein, in two adjacent cells in the same tissue, they can differ by 3 orders of magnitude. So it can be 1,000 times more of one particular protein made by the next door cell as the other cell, right? And so there's, like, in other words, there's tremendous stochasticity and variability on the tiny local level. But at the high level, you don't see, like, radically different formations in the organism as a whole, right? I mean, everybody has more or less the same size lungs, the same muscles in the same places, and so forth. So at a higher level, it's more stable. Biology is more stable, and at a low level, it's more stochastic. And so my argument is that proves that it has to be based on matching the outer conditions, which are more stable than the local conditions, which are very stochastic. Because if it was driven by the genome, then those two cells that are side by side would be expressing that protein almost identically. Does that kind of make sense? Okay.
I have a question that's sort of related to that, with because I understand this much better in the larger scale, just because I don't have a biology background. And so when I think of things like a murmuration, for example, which has where the starlings all follow really simple rules, which are fly towards the center, don't run into each other, fly the same, sorry?
Go away from predators.
Go away from predators and fly the same speed. The predators are the environment, right? And so I guess my question is around emergence and accumulation, where the instruction is coming from, whether or not that's like a genetic instruction to fly away from predators, or if that's an extra, that's where I'm a little confused.
Yeah, that is an awesome question. And it's like, try to think about a lot lately, and especially as I learn more about emergence, because I've been sort of looking everywhere for like alternative ways of solving this problem, or like alternative thinkers that, you know, have looked at this same problem and looked at it a different way, and especially in math. So Hector and I, who's on the call, Hector and I have been trying to build this mathematical object, which is a network of oscillators that sort of solves this maze. And we're trying to find a way that oscillations, it can be selected, you know, selectively, like pruning oscillations can eventually result in like a functional baby cell that solves a maze. And so we've been looking for like mathematical, you know, precursors, and that's one thing Sam has also helped me on. And my understanding of most of the like science of emergence and complexity theory is the idea is they're simple rules at the lower level that are sort of, you know, contained in some sort of particles. And then those simple rules, when they reach a certain scale, they create a different behavior, right? So they're consistent on the low level, they're precise at the low level, but then at a higher level, some sort of greater complexity emerges. And so it's really driven by these specific details that are totally precise. And then at a higher level, they become, you know, this more malleable structure. And so, you know, that's something like a fractal, right? So if you repeat a certain simple algorithm over and over, it generates these incredibly intricate, beautiful patterns, and they sort of resemble natural patterns in some ways. And so that seemed like that was a clue towards, you know, understanding biology, because you know, biology contains at least one particle, you know, which is the genome, which is very, very precise. And so I think that's this underlying assumption that our whole sort of interpretation of biology is based on. And I want to oppose that with like another, like, there's no question that the genome is precise, you know, but if you just view it as a toolbox, you see that everything else in biology is totally imprecise at a low level. Like there's all sorts of, you know, plasticity. And so accumulation is the idea that, you know, from the external conditions, somehow, maybe through oscillations or maybe through some other undiscovered thing, the interior of whatever system it is totally reformulated, and the details themselves are changed so that the details can be imprecise, but that the overall system can form a unit that matches its environment, matches its surroundings, and it sort of anticipates its surroundings, right? So knowledge is something that anticipates our surroundings. I mean, we have, like, you know, when we know something, it's sort of a prediction about it, right? We know what's going to happen next based on the context.
Yeah, I mean, that's all really consistent, I think, with when you think of a murmuration, it's a complex adaptive system, right, that is responding to predators. And so the pattern changes drastically if there's a hawk that swoops in. So I guess my question is where is that instruction? It seems like this downward causation explanation is that the actual instruction is coming from the environment. But could that instruction be embedded within genetics just on how to respond to the environment?
Well, there's no question that something is recorded in genetics, right? I mean, there's a reason why this molecule doesn't change very much over time. So it's definitely part of the system that's a critical part. But the question is whether it whether the system derives its organizational logic from that molecule or not. And I think that's what I'm trying to dispute, that the organizational logic has to come from the environment, and somehow the system is constituted in such a way, and that constitution, like how it's constituted, is still a total mystery, because we can't build a biological system from scratch out of non-biological stuff. If we could, we could say, if we could build a cell out of non-living material from scratch, then we could say, oh, we know how a cell works. But since we can't do that, we have to, I think, have to acknowledge there's a huge mystery there. And so if I have it right, then the cell is something that from the beginning of evolution, from the initial origin of life, was outward facing, was something that accumulated into its structure the external conditions, and a part of that accumulation is recording the solutions in the form of templates. And that's why, to me, it resembles symbolic communication in humans. Like, I think that the DNA templates, they're interchangeable units that can be combined in a way that creates meaning for the cell. And it's the same way that, ones and zeros, or A, B, C, and D, can be combined for us in ways that make meaning for our entire organism and our entire society. I drifted a little bit from your question, but
well, I'm just, yeah, I'm sure other people have questions, but I think the thing that I'm going to,
the question of whether it's a recording or whether it's a script.
Yeah.
And
and also whether or not, like when you scale it up, you can create a complex adaptive system. You just have to create a bunch of agents that interact with each other using simple rules. You can't create like life yet, but I'm just like, if it's the same rule that scales, like, could you do a larger scale thing in order to test this? Does that make any sense?
What can I dig into your question a little bit?
Yeah.
Because you said you can create a complex adaptive system if you just implement
But a bunch of agents. I mean, this is a complex adaptive system.
Yeah, no, but this involves cells. So this is like, we don't know how this is working.
Oh, I guess that's true. But I mean, if you yeah, I guess. But you can create, computer scientists have done these basically murmurations that online, which are just these agents that follow simple rules and interact with each other and create interesting patterns. None of them are alive, but that is a complex adaptive system.
Yeah, I'm agreeing with that. Well, I don't know if I would agree with the idea that it's adaptive. I mean, I think the adaptation that there has been put there by the programmer. You see what I mean? Like, the programmer fixed it so that system behaved in a certain way that matched something that they wanted to demonstrate, which was the flight of birds. You know what I mean? Yeah.
I guess, are the genes the programmer? I don't know. I mean, if it really does mimic exactly the same behavior and then creates this sort of unpredictable.
Well, that's the question. Are the genes the programmer? So if all that could be inherited was the genetic code and their epigenetic inheritance wasn't possible, then we would have to say, yes, the gene is the programmer. And that's the way everyone looks at it now. But I think that explanation is crumbling. Okay, someone put interesting comment. This is Dave. This conversation is making me think about the matter of inherited trauma. Barrie's really nice example of the murmuration. It's one thing to respond to a predator in the immediate moment, but it's another to respond to a predator that hunted one's parents or grandparents. This sort of trauma transmission is well explained already. But if so, I haven't heard that explanation. Yeah. Well, I think the study of trauma, well, I don't know that much about it, but it definitely seems to be bound up in this new discovery of epigenetics. That we're finding out that events like I think the most famous early example was there was a time during World War II where some population in Europe, I can't remember if it was Denmark or Sweden, somewhere there in Drew's neighborhood, underwent a period of starvation, and
It was the Netherlands.
the Netherlands, and children that were at a certain state of pregnancy of gestation inherited some epigenetic marking that then they passed on to their children, and so forth.
Yeah, it was right after the war, actually.
Yeah. And like all that is not a threat if it's just a one time and it's just like a random deviation that goes away. But if it's something that's adaptive and it can be passed on, for many, many generations, and of course we haven't had genetics long enough to know in humans, but in fruit flies, they can definitely make this go on for, 15 or 20 generations. And I think the understanding is that if it's adaptive, it can go on forever, right? If it's helping the animal survive, then it can go on forever, because it could just become the permanent new normal interpretation. And so that means it can happen really quickly. And so if genes were in control, they would have to mutate more quickly than that, to control those deviations.
Can genes be rewritten by epigenetic effects? Like, is there a feedback loop between the RAM of epigenetics and the ROM of genetics?
I think the answer is yes, but it's extremely rare, and it's like only in certain special cases. Like Barbara McClintock discovered the these transposons in maize, corn plants, and so that they're conditions under which, especially stressful conditions, under which some organisms can actually rearrange their genome. And of course there's horizontal gene transfer between bacteria, which is when two bacteria that are not closely related and they're normally asexual, they get together and exchange DNA. And then there's a whole book by James Shapiro called, I think, Evolution in the 21st Century, which details in mind-numbingly precise detail all the ways that he calls natural genetic engineering, which is organisms changing their genome under certain conditions. And then in the human body, there are immune cells that hypermutate to produce new antibodies. So yeah, cells can change their genome, but it's not the normal pattern. It's like very unusual. It's not the main way that epigenetics is inherited. The main way is that a new like group of methyl groups and like chromatin, and there's all this stuff around the DNA that controls expression. And so that stuff is what they call epigenetic marking, where that stuff is changed. And then those changes are inherited. And of course, that's the context for the change of the genes itself, when they randomly mutate. So there's no, yes, sir, Uncle Robert.
Charlie, do you know the beak of the finch?
Yeah, I've actually got that on my shelf. I've been working my way through it.
So how does explain how the changes occurred in the finch in order to stay alive over time? It's been decades since I read it, but my memory is that if the food was hard to get, the beak of the finch got longer. And this is studied over a period of years by some folks from Princeton, I think. But then when food became plentiful, the beak didn't need to be so long, and so the finch offspring didn't have long beaks. So that something is regulating that. I thought maybe you might be able to explain it.
Okay. Right. So So natural selection works on the cell just as much as it works on the genome, right? So if an organism is passing on things both genetically and epigenetically, then the epigenetic inheritance is not going to be slowed down any by natural selection. And so it's going to happen at least as quickly as genetic inheritance, but obviously it can happen quicker because there's all this there's so many different ways that the genes can be expressed, and there's so much that goes into, like, my analogy is, if the DNA was coiled up the size of a football, the rest of the cell would be the size of Central Park. So there's like this tremendous envelope of other stuff that's controlling the gene expression. And if any of that other stuff is changing systematically, that is that's part of the answer. I mean, obviously, if obviously it has to affect the germline cells. I mean, it can't be, changes that but there again, there are a lot of organisms that don't have, like Phil Marshall's favorite organisms, most plants don't really have a germline, right? I mean, they have, back me up here, Phil.
It is true that plants don't have distinct germlines in the sense that they're continuously embryonic, and so you can have new germlines arising from somatic cell lines, and so you get what are referred to in horticulture as sports. So there are mutations that can actually happen in cell lines within a single tree, for example, and so a particular branch will develop a new morphology or new pigmentation, and those are then sometimes propagated by horticulturists for ornamental purposes. So plants are sort of weird in that a lot of the conventional concepts that originate in zoology and human biology don't apply as neatly to plants, like the species concept, for example. But yes, they're plants don't have distinct germlines. They have multiple germlines arising from the somatic cell lines continuously.
So do sports ever arise that are not genetically distinct, but they're morphologically distinct?
That's a good question that I don't know the answer to. I think the assumption has been that if they are morphologically distinct, that they are genetically distinct, but I don't actually know.
Yeah. I mean, one of the things that's weird is that random mutation would be like an incredibly slow process. So like in the beak of the finch example, it's awfully hard to explain how random genetic mutation could happen quick enough that it would actually, systematically control just the beak length and nothing else of the bird, like within one researcher's lifetime, right? I mean, that was what the book was about. I haven't read the whole book, but my understanding is they studied these Galapagos finches for 40 years or so, and they found that the species was drifting within 40 years, had drifted significantly, in a direction that was adaptive. And so, the whatever this is, whatever the process is, whether it's epistolution or genetic change, it's not supposed to have any foresight. It's not supposed to know, I'm drifting in the right direction. And so random change, there's so many more, like just trillions of more ways that an organism could be configured that would be dysfunctional than ways that would be functional. And so if this was all attributed to the genetic sequence and random change, it would be hard. It's like, so in one of those lectures I talked about I think it was a book you sent me, Uncle Robert, about Stephen Jay Gould's debates with Dawkins about punctuated equilibrium, and there being these moments in evolution where evolution happens really fast, and then it reaches an equilibrium, and the ecology and biology doesn't change for a long time, for like 100 million years, and then something dramatic will happen, some disaster, and then there'll be another punctuation and it'll change again quickly. So I think that Beak of the Finch book actually is an argument in favor of epigenetic inheritance and genes being followers. Because if you imagine there's an epigenetic change that makes the organism more functional, well, then the genes are being selected, they're still being selected in the context of that, right? So let's say, I'm going to give an unrealistic example, but let's say, I live in an environment where I have to like do pull-ups to get into high trees and get fruit. And by doing lots of pull-ups, I get strong arms. And there's some tiny bit of that, gets epigenetically imprinted on the germline cells in my family. And the children are born with strong arms. Well, any genes that support that strong arm phenotype will be selected for, because that's our way of life at that point, right? As organisms, we're all doing that all the time. So, so at that point, there's a selective pressure to maintain that behavior in selecting the genes. And so I think that's the concept of genes as followers that's so that's powerful, because that way of thinking of it encapsulates everything we know about genetic evolution and explains all of that, and it just adds another layer of explanation on top of it that would explain why evolution could happen more quickly and like retain function better. Was that
Yeah. Sounds good.
okay? Yeah. Complex adaptive systems. Yeah. Okay, let's take another question. Who's got like an oddball question that's like not down the, I don't know how much did you guys, any of you guys have a chance to watch that first lecture? I was kind of I was focused on the first lecture was mostly focused on, I actually woke up in the middle of the night last night and I thought of a different way to organize this. But, to me, the evidence for epistolution is, first, differentiation that we talked about, second, epigenetic inheritance that we just talked about. But the third line of evidence that I didn't even mention in the lectures is the microbiome. And to me, that's one of the more like interesting examples where you have, so first of all, what is the microbiome? That's a tremendous number, many trillion single-celled organisms primarily that live in on and inside your body. And they function in a really surprising way. We've discovered in the past 20 years that they're composed, they're not your cells, right? So they're mostly bacterial cells. Uncle Robert does research on some of these little guys, the gram negative, if I recall correctly. And and some of Uncle Robert's research too is on the ways that the immune system is intertwined with this ecology of microorganisms. And so my understanding of this at a sort of higher level, definitely don't know the details nearly as well as a biologist would, but the big picture is that, you know, there are a tremendous number of these cells, and in each individual they don't have our gene, they don't share our DNA. They're foreign microbes. They come from the environment. We're born without them. We acquire them from, you know, early in life, and then most of them are in our gut, and then they form themselves into a community that's stable over our whole lifetime, like a fingerprint in a sense. I mean, they change when you eat food, so it's like, but then they regress to the same pattern. So it's like, you could think of it as a fingerprint on a rubber glove. So it stretches when you eat a certain food, they'll change, but then they'll kind of regress to the same, you know, combination of whatever it is, Prevotella and Bacteroides and Firmicutes, and, you know, there'll be some combination of species that live in you that will be unique to you, and you could almost use it like as a forensic tool to diagnose who's been where, because it's so unique. And so if this community of microbes lives inside us and it's interacting with our immune system, we're stabilizing that community somehow. You know, it can't be the DNA of the individual microbes that's stabilizing it. I mean, of course that's involved, but there has to be some outward effect that's creating a stable community there. And it's vital to our health. I mean, it functions like an organ. Like if you mess with your microbiome, your you know, your brain, you get depressed, or, you know, your immune system goes wrong. And so it's like vitally important, and yet it's composed entirely of foreign DNA. So it's really hard to explain how, if the instructions for life were in the DNA, how could that occur? You know, how could that sort of community be? Like, how could it behave itself? Does that make sense? Yes. Okay. All right. All right. Well, you guys
have you heard are you familiar with the term symbiogenesis?
Yes, totally. Yes.
Because I'm not a pro, but that term has made its way into literary studies recently, largely for me through the work of Anna Lowenhaupt-Singh and Donna Haraway. And yeah, that's just, I'm wondering if I think Anna Singh's book, The Mushroom at the End of the World, is a really lovely a book that has a lot of biology and anthropology in it, and makes it available to people who aren't specialists. Like it's a book I teach undergraduates, and it works out just fine.
I think you might have shared that with me. I think you might have shared some of that with me. That's awesome. Yeah, symbiogenesis is fascinating. That's the idea that organisms are created by combining in new ways, like ecologically, and then also, you know, it's the origin of the eukaryotic cell, is supposed to have been by ingesting another smaller microbe that then became like the mitochondria, and maybe some of the other organelles have their origin in an original, you know, combination of two organisms.
Chloroplasts also, in addition to mitochondria.
Awesome.
Which make green plants possible.
Yeah, it's totally fascinating.
Charlie, I excuse me. First of all, I'm very impressed by how smart your friends are. I won't say I'm surprised, but I'm delighted. And second, I just want to tell you an experience that I have that's is sort of relevant to what you're talking about. So I study a gene that is present in the horseshoe crab, and which means it's 450 million years old, and it's remarkably conserved. And the gene that the horseshoe crab has, which helps it deal with a bacterial molecule, we also have that gene. It's on chromosome 7. And for reasons that nobody understands yet, it is regulated by an intronic region on chromosome 6. Now, nobody can understand that. All my smart friends at the NIH have bombed on that. But it's so it's solid because people who have mutations in the intronic region on chromosome 6 produce less of this enzyme that's produced by the gene that I study, and they're more susceptible to have inflammation in the colon. So it's a very interesting and somewhat perplexing story that may actually involve something epigenetic. You may make a convert out of me by throwing this difficult research problem my way. Anyway, I'm very stimulated by your comments. I haven't quite yet become a believer, but I'm on my way. So.
Well, thank you. That's a high compliment. Very high compliment.
Charlie, what questions do you have about Epistolution? What's the stuff you haven't figured out, or whether rhetorically or scientifically?
Yeah, well, Hector and I have been like breaking our heads against, because I mean, the problem is that, you know, in science, really the way the game is played is you have to come to the table with some new evidence in order for people to pay attention to you and listen to your interpretation of everything else. Like if you produce some sort of dramatic new form of evidence, then they'll listen to what you say about everybody else's evidence and everything else. And so Hector and I have been trying, you know, for most of a year to produce this system of oscillators that is just my wild guess as to how accumulation can happen. And you know, I discussed it some in those lectures, but it's basically the question I'm trying to answer is how, you know, if the organism's instructed by the environment, then that has to be some sort of blind search, because we're assuming there's no, you know, divine creator instructing organisms what to do. So we're assuming that there's some sort of atheistic explanation for instruction by the environment that is biased towards function. It doesn't mean it's always functional, right? So if like you get, you know, if you inherit an epigenetic tendency towards obesity, you know, because of your family history, that's not necessarily adaptive, right? You know, so but we're saying, you know, if this is widespread, we're saying it's biased towards function, because otherwise it wouldn't exist. And so if that's happening, then you know, the organism has to be divided into units that can be selected between. There has to be some sort of selective process happening within a living body. Like something's being reinforced and something's being eroded by the environment. And what is that something, right? And we can just have noticed, like, for, you know, definitely since Lamarck, which was late 18th century, we've noticed that organisms respond to use and disuse. I mean, it's totally intuitively obvious, right? I mean, we exercise for some reason good for us, right? If you exercise a machine, it's bad for the machine. The machine breaks down more the more you use it, you know. But an organism, for some reason, when you exercise, you know, the structures and functions get more robust. And if you don't exercise, they deteriorate, or they, you know, sort of change. And that's like a general observation. We don't have any mechanistic understanding of how that happens and why exactly. Like, what logic is it following? Like, what is the environment? To me, it has to be a blind search, a selective blind search. And so the reason I'm using the oscillator as a concept is that that could be something that's selected between. So if you think of, chemical reactions as circular, as repetitive, and there are many of them nested within one another, the environment is selecting some circular chemical reactions and strengthening them and weakening other ones. And they might be nested together, right? So a great example is addiction, right? So you have this flow of chemical reactions that support addiction, and they go through the hormonal system and the neurons and even the digestion, and everything, becomes focused on sort of feeding the need for, caffeine or whatever it is. Your whole body anticipates getting your fix. And, that is just, almost ubiquitous, and it's so closely related to cognition in general. I mean, your expectations about the world, that apply to almost everything are kind of a version of that. The habits, we're habit forming, like we're objects that form habits. And so my idea there, and so Hector and I have been trying to somehow empirically test that by making this computer simulation of a network of oscillators that are selected based on use and disuse. And we're trying to make that into a structure that can just like solve a simple problem of like going into a maze. And so far we're failing. Like, I take total responsibility for that. But so that's a huge mystery. Like, it's the mystery, and I would argue it's the mystery of our era. Like, it's actually, that is the question that people don't quite realize that is like, if we can answer that question, then we will have like what I would describe as mastery of biology. Like, we'll actually be able to turn cells into structures that we imagine, that we want. We'll be able to create functions and that are adaptive in like a completely like freewheeling, unfettered way. I mean, and regeneration of particular organs that have failed in people that need them is just the drop in the bucket. That's just the very beginning of everything that could be done. And so, being able to fully control the cell environment and shape the fate of cells and what they turn into, what they want to do, their goals, and what they scale into, and of course ourselves, and there has to be a mechanistic explanation. I mean, there has to be some mechanism underlying it. And so, that to me will be the first artificial general intelligence. And so that's the whole theme of my website, Talking Octopus, because I think of it as something shaped like an octopus, because you'll have to have sensors and effectors, and you'll have to have some sort of computer that's running whatever this mechanism is that's undiscovered, that's accumulating knowledge of its context. So it'll have to have sensors that are going out and touching the world, and then bringing back that knowledge and integrating that into some sort of oscillating structure that holds knowledge. And so
that's a great place to end it for me, Charlie, because it's 6 p.m. here in Copenhagen, and I've got to go out for New Year's. So
right on, man. Thanks for joining.
Thanks. Yeah.
Awesome.
Happy New Year's, everybody.
Awesome.
Happy New Year.
Happy New Year.
We gotta go too, Charlie, I'm afraid.
Okay, well, thanks for joining, and
Good
greatly
to see you.
appreciated.
Nice to meet your friends.
Yeah. Great. Bye-bye.
Hello.
Yeah. Phil, you haven't said much. I'm so curious to hear your take on all this. Where are you in the
Well, I'll admit I haven't watched your lectures, but I probably should now. I really just wanted to hear what your current line of thinking was. So yeah, this is interesting.
Yeah.
Charlie, I've got a journalist's assignment for you to help. That is a question that I have that might help you connect what your idea is to the BS that everyone else is on about right now. It seems to me like one of the things you're interested in is a real artificial intelligence, as opposed to the not real artificial intelligence that is currently the way people are going about AI. The journalists are all in a Twitter about Chat GPT right now, which does some really remarkable things and is going to be clearly a disruptor of our lives soon, perhaps. And I think it would be really interesting for you to write about why that's not real artificial intelligence. And to use that as an opportunity to say, well, what would real artificial intelligence look like, and why, for all of its disruptive potential and usefulness, why a really, really complex language modeling system is not intelligence.
Yeah.
And because it sounds like that's what you're after, is you're trying to build a machine that will learn the way people learn, or octopuses learn.
Yeah,
if that makes sense.
Exactly. Well, and one thing that makes it like very kind of bittersweet and kind of no less important, but kind of bittersweet, is that the kind of thing I have in mind would not be able to answer questions like a magic genie like GPT can do. GPT is a tool that's much more powerful than the type of intelligence I'm talking about. Like,
you want to make a baby. You want to make an infant.
It's moral, right? Its superpower is morality, not like quickly solving search problems through Google, and using the whole internet to like answer, triangulate like a particular answer to a particular problem. It's knowing how everything fits together. And so, yeah, that's a really good assignment. Thank you. That's a great idea.
The other, now that you say it's, you reminded me about the conversations we've had about how questions of morality play into this, and that's not a journalistic article. That's like some big philosophical tome that just how to get from genetics to morality is I'm sure a lot of people have written about that, and I have not read those people, but I know I can imagine that lots of philosophers who think about morality don't actually communicate with scientific discourses with evolutionary biology. Well, maybe they do. I don't know. I'm sure they do, but I bet not well. Maybe you could do it well.
Well, I mean, you're totally right. I mean, there's no general theory of how morality emerges from biology, that I'm aware of, other than the sort of selfish gene, the arguments about altruism that come from, kin selection of genes and that sort of thing, which are pretty diffuse. I mean, everything has to be like, every explanation has to be compatible with genetic evolution. Right. I mean, but it's a question of whether it's sufficient explanation or not.
The connection, the story of the selfish gene and survival of the fittest, and like the way in which, I don't know, this is not a totally coherent thought. I think a lot of people, including Anna Singh, one of the books I posted in the chat, there's a sense in which certain rhetorics that have come from biologists, from Darwin to Dawkins, have had the authority to explain, to justify a certain kind of way humans are and should be. That is suspect. or I think, I'm not saying this very clearly, I think people are looking at Dawkins and to a lesser extent Darwin now, and saying like, what they wanted to see as nature was actually culture. It was actually like a human model. Like someone had read Hobbes and was projecting Hobbesian ideas onto that was conditioning why you choose the gene as the unit to look at, and not look at other things, not look at systems. And now I think the revolution has turned. It's gone from a kind of masculine single gene hero model to a network model, which maybe you can call feminine, or maybe you could call androgynous, or you could call masculine heroic, where the agents are not the selfish gene, but like the collaborative symbiosis of bacteria in your gut and the rest of it. What I guess what I'm saying is that it seems like our morality is influencing the way we talk about science, even as we are often reaching to science to justify our morality. So it's a weird, it seems so messy. It seems so messy. I don't know what I'm saying, really.
Well,
Just that it's an interesting.
no, I mean, you're totally dead on. I mean, I think like moral progress is the main question that I started trying to answer with all this, you know, is why does morality change over time? Why is our morality changing? Like if genes are instructions and our genetics haven't changed very much in a long time.
And if I think I would love for you to write something about
inventing these new ways of life that are more moral, right? We're inventing ways to be kinder and gentler.
The question that I'm going to just jump in here, because the question that David was bringing up, I think is important, and is basically you can look at the same behavior and describe it completely differently. And what you're like, you can describe it as survival of the fittest, or you could describe it as like search for knowledge and like, you know, whatever, coherence amongst all beings. And so that, I don't know how do you make that paradigm shift when it's the same evidence that you're looking at? Is that what you're getting at, David?
Wait, I'm not sure spit that out one more time. I'm still processing what you're saying.
So you were saying that like basically the moral framework, whatever, system that people bring to the evidence is that meaning is what they see in whatever evidence they see, right? So you can sort of look at evolution and say, oh, this is about survival of the fittest or the selfish gene or whatever, or you could look at it and say, this is a blind search for knowledge that will end with, you know, harmony amongst all living beings. But it's the same evidence that you're looking at. So yeah. What is it? How can you frame it? Like, what is it that you can do that would allow for people to see that paradigm shift, if that's the question?
I mean, one problem I have, it's a hard question. It's really hard. I mean, one thing that I thought would be helpful would be to point to the contradiction in our theory of life, you know, but like getting down to that contradiction is really tough, because there's a lot of like architecture in the way of it, you know, of like really imagining. I mean, all of the problem, this is a problem endemic to evolutionary biology, is that you have to just embark on these thought experiments. And it's so easy to disagree with a thought experiment because you're not talking about specific evidence, because no one is like witnessing very much evolution in like real time, you know? So you have to embark on these thought experiments, and you know, the one about epigenetic inheritance is pretty powerful, and also about differentiation, you know, and the knowledge that a different set of conditions can cause human cells to behave radically differently, you know, and how that corresponds with an instructional code in the genome. You know, it only fits very awkwardly. I mean, like one of the most exciting things is the discovery, like it's 2006, I think I was reading about this morning, but this Japanese researcher called Yamanaka discovered several genes that produce particular regulatory proteins that just 4 genes, turning them on forcibly, can make an adult cell turn all the way back into an embryonic stem cell. So it completely reverses aging all the way back to the beginning, where it's just like an embryonic cell. And of course, if you see that as a genetic instruction, it's put there like without ever having been like biology ever having really done it before, right? I mean, that's a completely novel trajectory for a cell to go all the way to adult and then all the way back to embryo and then all the way to adult again is a trajectory that presumably has never happened in evolution. And so you're like, well, why is that program even there? You know, how could that? It seems like we've got like totally the wrong mental model. If you see it as tools, then you can say, well, there's no end to the creative structures you can build and pathways you can express with a set of tools, right? Especially if it's a lot of tools. You could build any structure you can imagine and many more, you know, that you can't even imagine. So that's like how I imagine the future of biology is just sort of unlimited, like this just explosion of potential creativity. I guess that didn't really address the problem of looking at the same evidence in a different way.
No, I mean, it does in some ways, because that's a world I would rather live in. I mean, like the architecture metaphor, I think, is helpful in that, like, you know, if you have like wood and nails and whatever, and some like understanding of physics and some skills, and you can build whatever you want, given the things that you have, as opposed to just like the same sort of house over and over and over again. And obviously you would do it differently if you were on a mountain versus like on an ocean.
Yeah. I mean, if cells can't, if genes can be turned on and off, then, you know what I mean? Like if the genes in themselves had some logic that prevented them from being turned on and off in the wrong order.
Yeah.
You know, then that would make sense as an instruction manual. Like you do step one and you do
Yeah,
step two, and
I think my
sometimes
biggest
prevent step one from happening again. You know, like you go up a ladder, like 1, 2, 3, 4, 5, and by the time you get to 5, the pathway back has been blocked somehow. And so there's a progressive order, you know, and that would be like a genetic program. But instead, you could go to 5 and then jump back to 3 and go to 2, and that's what the Yamanaka factor experiment is showing. It's like none of this is fixed, right? Like if we just turn on certain genes.
But my issue, I guess, I don't know. I guess like you can't accumulate, if you're a butterfly egg, you can't accumulate into a human being. Like there is constraints. The genetic, like there are lots of constraints, aren't there? Like you can't emerge from one, like you, like I'm not going to turn into a bat. I can't. And neither will my child, because we're bounded by something.
Bounded by
and it's not the environment.
Bounded by what? I mean, I just wonder, like, I doubt, I can see that program, like I'm imagining some apparatus that like surrounds your cells and gradually over time, like through just the stimulation of your cells, it turns your cells back into a bat. Like that's like extreme, like that's a great example because it's so far-fetched.
No, but it turns me into a bat because I never was a bat.
Yeah, exactly. You never were a bat. That's why it's such a great example.
I think one thing I'm taking away from this conversation is that whenever
like, how much DNA do you share with a bat? Like probably almost all of it, practically 99% or something. Like, so we'd have to change a few genes in principle that exist in you and don't exist in the bat. But like the great majority of DNA that you carry is going to be shared by the bat. Right? And we got to change like the morphology of the cells. But we just presupposed that we understand now how to change the morphology of cells by affecting the environment that they're in.
So Charlie, if you can, I think turning Barrie into a bat is a tall, that could be like project number 2 or 3. Though I love anytime an animal has come up in this conversation, my like whole spirit has been like illuminated. Like I think use animals for the story. If you can shut off a gene that got turned on, you would be a bajillionaire, because I can't eat pizza anymore because my partner developed celiac in the pandemic. And my understanding of how that works is that you're genetically predisposed. There has to be a gene kind of hanging out in your genome that means that your intestines have the potential to completely lose their shit when gluten is introduced. And if you could figure out a way to just shut that off, you'd be absolutely rich, and no one would need to, and you'd have a really material cash out. Like I'm curious. I don't know if maybe your life's work will not like result in that, but if you could explain, if you could like light the way from how a certain understanding of epistolution can help people ask the right questions to get to the answer that could help us shut off a gene. Because my understanding of celiac is that it was environmental factors that activated it. Like,
Yeah.
Can you just figure out that system?
If a loop, there's some sort of loop that's sustaining it, right? I mean, all the cells eventually, like in our body, die and are replaced. So, I mean, there's some sort of loop that has to sustain itself in order for you to maintain celiac disease, right? I mean, so if we begin to understand those loops, like the baby step, okay, bats is like turning into a bat. That's like, graduate level over here, but like, just baby steps, kindergarten, like next 5 years or something, will be just trying to understand more carefully what loops are connected to what, right? I mean, and that's kind of what we're doing now, is we basically, my understanding of modern medicine is just you trace one particular molecule or factor or protein, and you see what loop it's traveling in, what turns it on and off. And that's your basis for the whole pharmaceutical industry, is either like degrading one protein or introducing, a new molecule and as a process, sustaining it. So, I mean, But obviously the process that sustains any disease etiology is going to be like this. It's going to involve millions of those particles. So, I mean, it's not just going to be one, and so sort of mapping what all is involved in what I would call the oscillation of celiac disease, what is involved in that loop and what are the most critical things to change. Is kind of like the next step, of like not just molecule by molecule, but like.
Well, yeah, I mean, that sounds like it's not like that's where we get from the world where we're working with thought problems to like empirical risk. Like, I don't know, what are the right questions to ask to move us forward in figuring out how to deactivate a gene that got activated by a context?
Yeah, I mean, in principle, you could just do CRISPR and change the gene already, right? You could just make yourself immune to celiac disease by like probably, CRISPRing your liver. Barrie, you gotta go.
I gotta go.
All right.
This was so fun. Nice to meet you all.
Happy New Year's. Yeah.
I should probably go as well, but this has been really enjoyable, Charlie.
Bill, thank you so much for joining. I'm so excited that you came.
Yeah, and we'll have to talk more about your possible upcoming move. So
Yeah, absolutely. Yeah, absolutely.
okay. Well,
all right.
See you soon. Bye.
See you later. Yeah.
Charlie, I should probably go too, but is there an upcoming move for you? Are you moving?
Yeah, this is to Vermont in the summer.
Are you moving?
We're definitely
moving to Vermont. It's done?
Yeah. Here's Hector looking, making his face known. What's up, man? Did you get a haircut, Hector?
Yes, but a long time ago. I mean,
Oh, okay.
when was the last time that we talked? I don't remember, but probably like right after that, I got a haircut. So yes.
Yeah. Awesome. Yeah.
Wait, Charlie, where in Vermont are you moving to?
Probably Burlington area.
Cool.
Is the thought. Yeah. So
Awesome. That's exciting. That is closer to, well, you know where I am. It's far away from Vermont at the moment, but I go back to Massachusetts all the time. So
yeah.
that's really exciting.
Yeah, man. Yeah, absolutely. Yeah, but it's great. Great to see you here. Thanks for joining.
Hey, yeah, no, it's really fun. And it's, yeah, it's fun to engage with you on this stuff in a group setting. Well, yeah.
You added a lot. Appreciate it.
Well, thank you. Here's to more discussions in the future.
Right on, dude.
All right, man.
Well, Hector, okay, see you
Yeah,
later, dude. Yeah.
See you, David. Bye-bye.
Hector, what did you think, man?
First of all, who were they? Well, I heard that some of them were like family and friends, but is
Yeah.
just that family and friends or researchers and so?
Still recording. I'm gonna stop the recording now because there's no need to.
Yeah.